So Manolis was trained at MIT and studied for a PhD in computational comparative genomics, which received the Sprouse Award for the best doctoral thesis in computer science. Manolis is known as one of the major thought leaders of the ENCODE and GTEx projects. He's been extremely active in the scientific community and is highly decorated with awards in engineering, biotechnology, computer science, as well as basic research, including a very impressive Presidential Early Career Award in Science and Engineering, an NSF Career Award, and an Alfred P. Sloan Fellowship. His impact on the scientific community is remarkable, having published over 90 papers that have been cited over 37,000 times. Manolis and I often bump into each other pushing small children in strollers at genetics meetings, and I'm absolutely grateful that he's able to visit us today. So please join me in welcoming Manolis.
Thank you. I have to admit, I had one of the most awesome days. So at the lab, I bounce ideas of my students, and it's just wonderful. It's an amazing environment. But speaking with the students today was like flying sparks all over. I mean, you have some of the best students I've ever met, I have to admit. So you guys are truly awesome. Of course, the faculty were up to par, just to match the amazing students. So what I'd like to tell you about today is our adventures on using gene regulation as a lens to understand human disease in order to reveal the circuitry underlying many of the top disease hits, and using that circuitry to then guide manipulations to often reverse the disease phenotypes. So what I'm going to talk about today is how do we now take this wealth of data that has been generated by both the genetics community on the GWAS side, as well as the functional genomics community on the epigenomic side and the regulatory genomic side, and sort of put it together to gain insights that would not be possible with either of the two worlds alone. And I'm going to start with the promise of genetics for human disease. You see here one of the folks sitting actually in the room, Speliotis, Liz, that you know all very well, basically carried out these studies of BMI through the years. And the first and the most, the strongest, and still to date, the single strongest hit, reigning above all the other BMI hits, was in this FTO region of the genome that has basically suddenly shed light into a part of biology that was previously not recognized. But from the discovery of that association back in 2007, when this hit first came out, to the understanding of the mechanism of this association, it took actually more than 7 years to basically understand how do we translate what was discovered very statistically reliably from genetics alone to a mechanism to actually translate it. So that's the challenge that we have. We want to start from genetics and understand disease mechanisms, understand new target genes, develop new therapeutics, and enable personalized medicine. But the challenge there is that it doesn't stop with the genetics, it starts with the genetics. And the difficulty is that when you open up the hood in these regions of association, and here's that FTO example, you basically see that there are dozens of genetic variants that are co-inherited, that are in very strong linkage disequilibrium, and are impossible to resolve using genetics alone. So you don't actually know what the causal variant is that's driving this particular association, or what the set of causal variants is. And that is not as much of an issue when you have a protein-altering variant, because you can make the simplifying assumption, which will sometimes be incorrect, that that protein-altering variant is the driver. But in this particular case, there are no protein-altering variants. It's all non-coding. And this is not just some weird case. It happens in 93% of GWAS hits. In 93% of cases, you basically have these common variants that are associated with disease, but there are no protein alterations in sight. So something changes in the circuitry, we assume, that will somehow change the expression, maybe of this gene that's harboring the genetic variant, maybe of some other gene nearby, or sometimes very far away. And in this particular case, it turns out to be 2 genes that are 600,000 nucleotides away, and 1.2 million nucleotides away, that are not by any stretch of the imagination near or even the adjacent genes. So how do we translate now these genetic hits to identify, number one, the target genes through which the genetic variants are acting, Number two, to identify what are the causal variants. And now to utilize these causal variants to understand what cell type these regions are acting in. Because the power of genetics is also the curse of genetics. The power of genetics is that you don't need to know the mechanism in order to discover a genetic association. The curse of it is that once you've discovered the association, you don't know the mechanism. You don't even know where in the body it acts. You have a genetic variant. It could be acting in brain, it could be acting in liver, it could be acting in adipose or in fat, or you name it. And that's where the mechanism has basically remained impervious to analysis in many, many of these loci. And I can count in one hand the number of loci for which a non-coding variant has been connected all the way to mechanism. And what we'd like to do is change that paradigm, just make it, tilt the balance to do that systematically for thousands of regions. So how do we enable that? The way to do that is to actually systematically understand the circuitry of the human genome in order to then utilize it in every one of the cell types, in every one of the disorders. And that's what large-scale projects like the ENCODE project, Encyclopedia of DNA Elements, or the Roadmap to Epigenomics project have set out to do. They basically said, let's build a resource. Rather than focusing on any one disease or any one locus, let's build a resource for every cell type that we can get our hands on, every tissue we can get our hands on, in adult as well as embryonic stages of development. And let's simply start dissecting all of the non-coding elements in one swoop and start building networks, linking these non-coding elements that are very often distal regulatory elements known as enhancers, linking them to the regulators that control them in trans, as well as to the target genes that they control in cis, and developing new methods for utilizing these annotations to understand disease. So that's what we want to do. We want to basically build these maps and use them to turn the tide on disease and go from association to mechanism. And the 6 things that we would like to have are, number one, understand what is the relevant cell type, what are the target genes, what are the causal variants, who are the upstream regulators, what are the relevant pathways, and what are the intermediate phenotypes through which that region is impacting phenotype. Everybody with me? So that's what I'm going to talk about today. So basically, number one, how do we start with these regions of associations? And I've pushed that button. We'll see when the slide actually changes. It'll be a very interesting talk. I've also pushed the button physically. So if anybody has a laptop. Yeah, but I don't want to do this all talk long. So I'm going to give you my USB stick, and if you can load it on your laptop. Can anybody volunteer their laptop while we're speaking here? Thank you. I'm going to let you load up. I'm going to give the outline, and when you're ready, we're going to plug yours in. Do you have a VGA adapter? So this is what I'm going to talk about, basically. So number one, how do we characterize this epigenomic landscape? Number two, how do we use it to identify what are the disease-relevant tissues and regulators? Number three, can we actually utilize what we've learned about the relevant pathways and the relevant genes and regulators in non-coding regions to actually prioritize new target genes that would otherwise not be visible using current cohort sizes? And I'm going to talk about 3 different approaches for doing that, using epigenomic signatures, using network convergence, and using epigenetic variation across individuals. Then number 4, once we have identified these regions, how do we go about manipulating them? And there I'm actually going to use the example of the FTO locus that I introduced at the first slide. And then how do we now scale it up? Can we develop new technologies for doing this systematically across the entire genome? So I also just clicked. So the first part is basically a collaboration between a very large number of both computational and experimental labs. This is all of us on my roof deck here. You can see my first genetic experiment. I have 2 now. They're both very successful. But still early on, we'll see how they pan out. But basically what we try to do in this particular, thank you so much. I very much appreciate it. My virus is now copying all your data during the talk. It's going to be shipped to Boston. So basically, what this project actually did is actually map the epigenomic landscape using 3 types of modifications. Number 1, DNA methylation, which is directly on the DNA, and it's usually associated, and feel free to change anytime, it's usually associated with silencing of regulatory regions, but it's also associated with activity in transcribed regions. It's a little weird. Number 2, the accessibility of the DNA itself for transcription factors to bind there and regulate whatever region of the DNA is here, both activating and repressive. And number 3, and by far the most complex, the post-translational modification of the histone proteins that hold DNA together. So these serve both structural roles as well as informational roles because, number 1, they hold the DNA together, but number 2, the modifications on them help interpret different chunks of the DNA in different colors of chromatin or different meanings. And there's 147 nucleotides wrapped around every one of those nucleosomes and about a 50 nucleotide linker. So you can think of them as 200 nucleotide chunks on the genome that basically have specific classes of functions. So what we did in this project is we gathered more than 100 different adult and embryonic tissues, individual iPS-derived cell lines, as well as ES lines, and several primary cells. And for each of them, we mapped a large number of these histone modifications, DNA accessibility, DNA methylation, as well as what you can think of as the output of RNA expression. So how do we put all that data together? Ready to switch? He's great. He's already gone to the right slide. Let's see if it works now. Cross your fingers, everyone. Max saves the day. Let's see. Ta-da. Yay! Woo! All right. Let me plug mine in. Are you running off the thing, or did you just copy it on the? Okay. I'll use it. We're good. Woohoo! Thank you. Thanks so much. All right. So how do we interpret that data? So this is what the primary data looks like. So you basically end up with dozens of tracks in some of the most deeply profiled cell types. This is the example of IMR90 lung fibroblasts that have more than 30 different epigenomic tracks. And you can see here, every one of these histone modifications is mapped across the genome. You can see the intensity with which that particular region of the genome gets pulled down with the chipstick experiment, as well as the amount of methylation in that particular region, the accessibility of that particular region, and the level of RNA transcription in that particular region. So what do you see? You basically see that this gene right here, PAX5, is actually quite repressed. You see these polycomb-associated repressive marks. You see these heterochromatin-associated marks, and this reduced methylation in that region, which is kind of interesting. And again, as I mentioned, methylation in transcribed regions is actually associated with activity. So less methylation is less active, whereas in regulatory regions, more methylation is actually repressive. And you can also see that there's a lot of correlation between different marks. So it's very difficult to interpret all that. So what makes an enhancer region is actually a higher level of H3K4ME1, but a lower level of H3K4ME3. Even though you see it here, it's at a lower level. And it's the difference sometimes between these marks that's actually more informative about what's the underlying state of the chromatin. So what we want to do is take these tracks that are genome-wide, and that are multiple, and that are complex, and where combinations actually matter, and translate them into a single annotation track that basically tells us what is the function underlying this in this particular cell type. And we'd like to have fuzzy names like enhancer, promoter, transcribed, repressed, rather than the individual marks. And the way we do that is that we run a multivariate hidden Markov model. The hidden Markov model basically says you have some Markovian process running along the genome, which is memoryless but depends only on the previous state. And you have a set of variables that are observable, or emission variables, that are the only thing you have access to. And behind it all lies the hidden state of the chromatin. The biological state, which is not directly observable, is hidden. It's only inferred. And that's the reason for the hidden Markov model. So we basically run this multivariate Markov model, which emits a whole vector of activities, to figure out in an unsupervised way, without any training set, what are the classes of chromatin in the genome? What is the language of the epigenome in some way? And it comes back with about 15 different combinations of histone modification marks that occur at very different frequencies across the genome. This one here, absence of these marks, occurs at about 70% frequency. And this one here occurs for only 0.1% of the genome. So it basically partitions the genome in very unequal ways between these different regions. It doesn't just split it up equally. And the most important ones turn out to be enhancer regions, which are presence of these marks but absence of these other marks. Promoter regions, which is, again, presence of that mark but absence of the other ones. Some of these bivalent regions. And you can see why am I calling them important, because they are actually more frequently evolutionarily conserved across different species. So if you look at the evolutionary conservation of these regions, again, that information didn't go at all into the model. You can see very different patterns for different regions, taking out, specific important elements. You can also see that, again, these data that didn't go into the prediction algorithm basically distinguishes repressed genes that are typically associated with this bottom half of the state, and these active genes here, which is associated with the top half. And that allows us to now start giving names to these based on their enrichment in different classes of elements, such as CPGs, exons, introns, transcription end sites, transcription start sites, DNF genes, and so on and so forth. And we can go back and start annotating that genome with a much more interpretable alphabet. So now we can ignore the complexity underlying all of these different data sets and start focusing on this single annotation for that cell type. That basically tells me that there's a repressed region in gray on top of this PAX5 gene. There's this active enhancer region, on top of this particular region. There's a lot of different promoter regions in red, and so on and so forth. And we can take this track now for one of the cell types and show it in the context of all of the other cell types. So I'm now going to show you 127 different epigenomes all on top of each other. Okay, so here's that top track, and you can see the PAX5 gene, which is repressed here in IMR90 fibroblasts. And you can also see how that region changes across different cell types. It's kind of cool, right? You suddenly have a very different view of the epigenome and its dynamics across cell types. You can see here that this region is repressed in most of the cell types, except for a handful of places where you see the promoter turning on, the gene is transcribed from the right to the left. You see these enhancer regions turning on, and so on and so forth. You also see this other gene where, it goes on and off in different groups of cell types. And what's really interesting is that the promoter region for that gene is actually constitutively on. The promoters seem to be a little more boring because they're, on whether the gene is expressed or not, at least according to these transcription models. But enhancers in yellow here are much more dynamic. So they turn on and off in very specific cell types. So we can actually take now all of these enhancer regions together and start linking them to their target gene. So if a particular enhancer region turns on exactly when a gene turns on, you can somehow link these together because there's correlated activity. And you can also start linking and grouping together all of the enhancers that turn on in similar ways. So basically here, there's this enhancer that turns on here, that enhancer that turns on there. We can basically group them into a brain class of enhancers that turn on across the brain. And we do that not just for this region, but we do that across the entire genome. And there are 2.3 million of these regulatory regions that we call enhancers that are acting at a distance. Let's cluster them all together. I'm going to show you now 2.3 million of these enhancer regions across 127 cell types. And I'm going to use half of a quarter of one slide. You guys ready? So here's a quarter of a slide. And now half of it is the 2.3 million regions here, and then the 127 cell types here. So again, by clustering the data, it actually starts making a lot of sense. You can basically see here that T cells and B cells have, one class of enhancers that's quite specific. You can see brain has another class here. And what's really interesting is that if you look at the genes nearby, you basically see that there's specific gene ontology enrichments for immune functions in T cells and B cells, for learning and memory, for brain, for heart functions in, this mesenchymal lineage here, and so on and so forth. So it's quite remarkable. There's like modules of enhancers that are scattered across the genome but are turning on and off at the same time. And that's really exciting because now we can actually look within them to find sequence patterns that might correspond to regulators that control these cell types. Because we have a module of enhancers that turns on again, on and off at the same time, we can actually look for common patterns that are overrepresented within them and get some sense of who are the regulators that control that module. And sure enough, we find a lot of the well-known regulators as well as a lot of novel ones in specific tissues and combinations of tissues. But we can also go the other way. As I mentioned earlier, we can look at the correlation between the activity of an enhancer region and the expression of nearby genes and start assigning enhancers to the genes that they might control. And we can complement this data with high-C data about the physical proximity of different regions of the genome based on how often they interact when you chop up the DNA and you religate it. You basically see how often have they ligated together. And you can, of course, also use expression QTL evidence of genetic variants that are correlated with the expression level of a particular gene at a distance, enabling you, again, to sort of see connections between these SNPs in a particular regulatory element and the expression of those genes. So we've gone through this computational step to build the circuitry of the genome, and we can utilize that to go back to these genetic regions of association and ask, what does the circuitry tell us? So we now look at one of these regions that's quite simple. It only has 4 genetic variants. And you can see now where are the enhancers that overlap these variants. What cell types are they active in? Who are the regulators that are predicted to target them? Who are the genes that are predicted to be targeted by these? And even in this very simple region, you can see that if I predict this particular regulator to be the driver, then I predict this enhancer to be driver, this causal SNP to be the driver, and this target gene to be the driver. But if I instead predict this particular regulator here, then it actually changes the target gene that I predict, and it changes the SNP that I predict. So there's a lot of interplay between what hypothesis you make at the regulator side, at the enhancer side, at the cell type side, or at the target gene side, and all of them are actually informing each other. So we can actually utilize that computationally to start iterating through and predicting what are the, move the slide further on. What are the regulators and causal variants and so on and so forth, okay? So we can actually do a lot of this computation now that we've put together that network. But we can also simply put all the data out there for people to start utilizing these predictions. So one of the things that we've done is that we've built this system called HaploReg for haplotype regulation that allows you to look at your favorite variant that's associated with a particular trait in the context of all of the other variants that are sitting on the same haplotype, and you can see the R² values here on the order of 0.99. So any of these variants could be responsible for this particular association. And for each of those, you can now start drawing hypotheses about what tissues they might be acting on, what regulatory motifs are disrupted, what promoter regions, what DNA hypersensitive regions, what conserved regions are potentially disrupted by these variants. So any questions so far? So here we've done the first part of basically characterizing the genomic landscape, mapping non-coding elements, uncovering their circuitry, and linking SNPs to the enhancers that they overlap, the target genes that are predicted to be controlled, the regulators, and the cell types in which they're predicted to act. And we could stop there. We could basically say, listen, we're a computational group. We're just going to put it all out there for people to use and go home and be happy and work on something else. But instead, we basically said, hey, if we eat our own dog food, maybe we'll actually start tasting better. So if we basically force our predictions to be good enough that we ourselves will follow them up, it will sort of set a higher standard for when something doesn't make sense, we'll try to improve it. So that's what we did. We basically said, can we start gaining some insights about these disease regions? The first is, can we identify disease-relevant tissues and regulators? And we've developed Bayesian methods for prioritizing them, and we've applied them and followed them up experimentally in specific cases. So how do we use these epigenomic maps to predict disease-relevant tissues? It's very simple. We basically start with all of the genetic variants that are associated with height. And we basically have the LD blocks but we also have the specific p-values for every one of the SNPs within these blocks. And we have this for many different traits. And why is it important to use multiple traits? Because we can actually use the different traits as controls for each other. Because any one trait has a lot of biases in terms of where are the LD blocks longer, where are the genetic variants at higher frequency, and so forth. But if we use multiple traits, we can actually control for inherent biases in the genetic variants that are typically disease-associated by playing off each other. And the different traits have, in fact, very different regions that they're associated with. So we can actually start mapping now the enhancers that we paint in each of the different cell types and see what are the tissues that are the traits that are the most enriched in the enhancers acting on a particular tissue. And you can see here that somehow height lights up for stem cells. And if I look at immune enhancers, again, and give everyone the opportunity to light up, but the strongest enrichment is found for type 1 diabetes. And if I look at heart enhancers, they overlap blood pressure-associated variants. And if you look at the genetic variants associated with cholesterol, they're enriched in enhancers that are active in liver. So I can basically start putting check boxes in this diagonal, which starts telling me that different genetic variants associated with different traits act in enhancers that are active in different tissues. So I can start mapping traits to cell types and tissues through the genetic variants that they're both linked to. And this is what the real data looks like. So again, if you look at height here, you see that there's no enrichment in any of the cell types except for embryonic stem cells. If I look at any of these immune traits, you basically see an enrichment in T cells and B cells. If I look at blood pressure, you basically see there's nothing much except for one tissue here, and that's the left ventricle of the heart. And what does that tell me? That basically tells me that there are known coding variants that are sitting in enhancer regions that are active in the left ventricle, where the genetic variants disrupt the enhancers, not the genes directly. And that ultimately leads to the phenotype of differences in blood pressure. If I look at fasting glucose-related traits, you basically again see very strong enrichment specifically for pancreatic islets. If you look at cholesterol, you basically see one very strong enrichment specifically for liver. This makes a lot of sense, right? So we would expect cholesterol to act in liver. What about inflammatory bowel disease? Well, you see these massive enrichments here for T cells and B cells. Makes a lot of sense. It's an autoimmune disorder. And you see this other enrichment here for digestive tissues. So inflammatory bowel disease makes a lot of sense. It's both the immune cells and the digestive system. What would I expect for Alzheimer's disease? What would be your guess? Brain, right? So I basically go and look at brain. This is brain right there. And I don't see any enrichments for brain. The brown box is brain. So there's something wrong. So either our method is wrong, or perhaps our assumption is wrong, that it works in the brain. What does the method actually tell us? It tells us that there's some weak enrichment here for various B-cell-related hematopoietic stem cells, but the strongest enrichment is for CD14 plus cells. And that makes a lot of sense if you actually look at a lot of the recent literature on Alzheimer's disease. So Alzheimer's is basically a little bit of an outlier here. We're going to come back to that in a second. But you can see how we can actually start clustering traits in specific cell types. And most of these make sense. What about Alzheimer's? It just so happens that Li Huitai, the director of the Picard Institute, works just across the street from us. And she has a mouse model of Alzheimer's disease that undergoes the same neurodegeneration pattern as the human brain does, but in accelerated time frame. So we can actually start looking at what are the epigenetic changes that happen during progression of Alzheimer's disease, which are different between control mice and the CKP25 mice that are the model of Alzheimer's neurodegeneration in mice. And we can profile what are the epigenomic changes that happen early and late. And here's what's really interesting. We find both neuronal processes get repressed, exactly as you would expect, but we also find that immune processes get activated. So this makes a lot of sense. There's basically inflammation associated with neurodegeneration. And a very simple-minded view would be that, yeah, sure, neurodegeneration happens, and that causes inflammation. That's normal. But what's really funny in this data is that the immune activation happens in the early time point here, whereas the repression of the neuronal processes actually happens in the later stage. So that suggests somehow a reverse causality, that somehow changes in immune processes precede the changes in the neuronal processes. And we can actually start mapping across all of the different tissues what is the correlation between the changes that we see in the mouse and the genetic enrichment that we see in humans. And what we find is a near perfect correlation between the two, namely the enhancers that go up in mouse are also the ones that are active in human AD. But that's not true for the decreasing neuronal processes. There's almost no correlation. There's also an anti-correlation. And in fact, indeed, neuronal cell types are the least enriched of all of the cell types in the genetic variants that are associated with Alzheimer's disease. And this indicates that immune cell dysregulation is in fact the causal component of Alzheimer's rather than just simply a consequence of neuronal changes. And there's two types of cells that are quite interesting here. The first is microglial cells that are CD14+, which are the resident immune cells of the adult brain. And these are both the cells that clear out the debris inside the brain, as well as the cells that trim down neuronal connections that are not utilized. So dysregulation of the microglial cells might in fact be a causal component of Alzheimer's, shifting the therapeutic focus from the neuron to the immune cells. It could also be the macrophages that infiltrate the brain during neurodegeneration, either helping out with the clearing out or potentially causing damage and neuronal pruning. So this completely unbiased view is pointing to a mechanism that's very subtle and very complex and suggesting that perhaps the genetics is actually telling us a very different story from a very simple-minded view of first neuronal loss, then neurodegeneration, then immune and inflammatory processes. So we've done a lot of additional computational work in the Bayesian prioritization of driver tissues, variants, regulators, and target genes. So we've now developed a fully Bayesian model that allows you to now start working with HaploReg in a more interactive way that allows you to sort of start formulating specific hypotheses and following up these hypotheses by prioritizing genetic variants and genes. And in this particular case, we've run in a fully automated way that allows us to predict what are the causal indicator variables for a particular variant in a particular disease that tells you how likely is it that that variant is in fact the driver for that particular association that's observed based on the enrichment of that particular annotation in that disease and the specific annotation of that variant in the tissue of interest. And you can now go to regions where a lot of different variants are stuck at the same significance level and apply this filtering approach, which basically points to one causal variant that's a likely driver. And in this other region, very many variants. Again, it prioritizes two, one of which is in fact EQTL. And you can actually see that at the larger scale, where you can basically see that the variants that are prioritized by this particular Bayesian framework are enriched in evolutionarily conserved elements, far above and beyond what you would get with other methods. And they're also enriched in EQTL from GTEx, far above and beyond what you would get from GWAS alone in the blue line here. So basically, all of this is suggesting that we can somehow use this iterative framework that I was alluding to earlier to start prioritizing the causal variants. And you can basically run an expectation maximization algorithm using the network connectivity that I described earlier to sort of prioritize what are the driver regulators, what are the driver variants, what are the driver cell types, and what are the target genes through this iterative procedure. And when you apply this to, in this particular case, total cholesterol, you basically see these large number of master regulators being predicted to be targeting a large number of target genes. And you can see here in star the predicted master regulators, as well as in orange the previously known master regulators. And you can see that our network is in fact capturing a lot of those. So that's about utilizing these insights about this network connectivity to prioritize both regions and tissues and regulators that are likely to be drivers. But we'd like to now not only use that at the large scale level, we'd like to also use that at the single gene level to prioritize additional genes that might become powerful new targets in the future, but might have to wait 10 years or so before the GWAS cohorts become large enough to be able to discover them using genetics alone. So we'd like to now combine epigenomics, epigenetics, and these regulatory networks to discover new loci that do not reach genome-wide significance, but that are biologically meaningful and indeed phenotypically relevant when perturbed experimentally. So we're going to use three approaches. The first is epigenomic signatures. So this is a very simple idea. So this is work that Xin-Chen Wang did in co-advising role with Lori Boyer at the biology department at MIT. Well, he basically said, let's learn combinations of epigenomic features in these genome-wide significant regions to start prioritizing additional genetic variants in these subthreshold regions that are biologically meaningful. And in particular, he focused on the QT heart repolarization interval, which is a very well-studied trait that has a large number of above significance variants. So he trained a machine learning model that picked out several features that are more enriched in the GWAS variants than what you would expect by chance, giving us several significant enrichments that can now be combined to prioritize subthreshold variants. So we're learning a set of features, and now we're going to apply those to the subthreshold variants. So he's able to now double the number of loci, and these are some examples that he validated experimentally, where he basically carried out a luciferase reporter assay to show that indeed that genetic variant is affecting the level of luciferase that you see in these enhancers. And you also are able to predict the target genes using a 4C experiment that's basically asking who is actually interacting with these enhancers and what are the predicted target genes. And what we find is that most of these are in fact forming long-distance enhancer-promoter interactions found out experimentally to nearby genes that make a lot of sense biologically. And now if you go and perturb these genes in zebrafish, what you find is that there's indeed a significant change in the heart repolarization duration for two of these candidates. So most of the experimentally tested ones indeed showed a change. But you can actually also use human genetics to basically ask, are these genes that are prioritized by the QT interval, subthreshold loci, also significant in the QRS GWAS? And the answer is by a very large margin and quite significant. They're much more strongly enriched. And you can also carry out a knockout experiment in mouse and basically say, do I see a mouse phenotype when I knock out these genes? And a lot of these mouse knockout experiments have already been carried out. So we can ask, are the genes that are predicted to be targeted by the subthreshold loci more likely to show a contractility phenotype or cardiac conduction phenotype in mouse when knocked out? And the answer is, again, overwhelmingly yes. So what's really interesting here is that we can discover these using GWAS cohorts that are relatively small right now. And we can estimate that you would need roughly 3 times the size cohorts in order to be able to discover those using genetics alone. So somehow we can be a little bit ahead of the curve in terms of starting to carry out experimental validation for these additional loci. The second line of evidence for prioritizing these new genes is basically looking at the network connectivity. And this is particularly relevant for rare variants that are occurring in very few individuals that would not become genome-wide significant using existing cohorts. And the extreme case of these rare variants is in cancer, where you basically have new somatic mutations that are different in every patient, and they're sometimes different between different cells in the same patient. So what we asked is, do these rare variants and these somatic variants cluster in the same regulatory network? And this is work by Richard Solari and Nick Armstrong. So heterogeneity is a huge, huge issue in cancer, where there's a very, very large number of new genes that are coming up and that are all scattered throughout the genome. But Richard's idea was, let's actually start looking at the connectivity of these scattered regulatory regions to promoters of potential target genes. So for every gene, he's building what he calls the regulatory plexus, which is the set of regulatory elements annotated with a chromatin state that are connected to the same gene based on experimental high-C evidence. And then he asked, is there an abundance of a specific gene and a specific set of enhancer elements targeting that same gene that is above and beyond what you would expect by chance? So to make a long story short, he basically finds that a large number of genes are dysregulated in prostate cancer, and specifically they're upregulated rather than downregulated. And those upregulated genes contain an abundance of rare mutations and somatic mutations in their regulatory plexus, in the regions that are connected to. And these regions that are connected are specifically promoter regions and enhancer regions by a very strong significance. And what's really interesting is that they're enhancer regions that are active in other tissues. So somehow what seems to be happening is that the prostate cancer cells are in fact co-opting enhancers that are normally off in prostate, but on in other tissues. They're de-repressing them, and they're utilizing that to turn on genes that would normally be off in that prostate. So we can actually use the statistical model to start prioritizing these genes by correcting for three things: the mutational frequency of the different regions in the genome, the mutational frequency of different chromatin states in the genome, and the mutational frequency of different patients, because all of these vary. And we've used the statistical framework to correct for all of those. And we can now start discovering new genes that do not contain protein alterations, but instead contain regulatory alterations. And why are these so important? Because it allows the cancer to co-opt genes that are facilitating the cancer, whereas most protein coding alterations are disrupting a gene. These allow you to actually overexpress a gene, as we showed in the previous slide. And what do these genes do? Turns out that they actually play very similar roles. So in the case of prostate cancer, you see that many of them actually play roles in insulin androgen signaling and spermatogenesis. They play roles in immune evasion, and they play roles in mitochondrial activity, which are, again, part of the hallmarks of cancer that Bob Weinberg elucidated, I think, almost a decade ago. So some of these regulatory plexi are, in fact, very complex. Others are very simple, but they all have a similar property that they're converging onto the same target gene. And these are methods that can now be utilized across the spectrum of variation, not just limited to the common variants, but rare variants and even somatic variants can be now clustered through this regulatory network connectivity to the same target gene. So we experimentally validated a handful of those loci. We basically found that indeed the long distance interactions are supported by 3C evidence. We found that these genes in fact fall in common pathways with other prostate cancer genes that make a lot of sense biologically. And we found that overexpressing these genes experimentally in a prostate cancer model indeed reduces ERK and AKT activity, which is synergistic with E10, which is one of the top loci for prostate cancer. So the third line of evidence for prioritizing these subthreshold loci is epigenetic variation. So we can now not only look at genetic differences between individuals, but we can also see if the epigenome of these individuals varies from cell type to cell type and from individual to individual. So we can now start complementing the, and somehow bridging the gap between this genetic link all the way to phenotype with what is actually happening in specific tissues, what are the regulatory alterations that are happening, what are the gene expression patterns that are correlated with that, and what are the intermediate phenotypes, and start tracing somehow the link to the disease. But there's a challenge here, which is that we lose the causality that we had for genetics. Basically, these unidirectional arrows no longer apply because the environment can be controlling any of those, and any of those can actually be downstream of the disease itself. So how do you deal with that? Well, we try to do that in the context of Alzheimer's disease in collaboration with Phil de Jager and David Bennett. This is an incredible data set, the ROS-MAP cohort that is gathered by David Bennett at Rush and profiled in collaboration with Phil de Jager at HMS, where the patients have been followed for more than 10 years with cognitive evaluations, and they donated brain samples post-mortem where we actually profiled both genotype and methylation. So we have 723 individuals for whom we have measured DNA methylation in the dorsolateral prefrontal cortex, which is associated with cognition. And we basically asked, what is the relationship, what is the interplay between genetic differences, phenotypic differences, and now epigenetic differences between individuals? And up until now, we've used the epigenome as a reference epigenome without caring about its variation across individuals, whereas we now are going to be looking at how it changes from person to person. So we're going to do three things. First, relate the genome to the epigenome and learn a mapping between genetic variants and the epigenetic marks that they affect, and that's simply discovering methyl PTLs. We're also going to learn what is the relationship between the epigenome and the phenotype in the context of a methylome WAD association study rather than GWAS, it's an MWAS. And then we're going to combine the three together to evaluate whether this epigenetic alteration was in fact causal by connecting all three. So in the first part, looking at genetic variants that affect methylation, you can basically see here the genotype on one axis and the methylation level in another axis. And you can see these beautiful correlations across the whole spectrum. And a correlation with genetic is usually a causal relationship. So that suggests that genetic differences are in fact driving methylation differences in these regions. What are the regions that change the most? Turns out that they're the intermediate methylation regions. You don't see any changes from these very strongly methylated or the strongly unmethylated regions shifting to each other. You instead see small changes shifting within these intermediate methylation regions. And these are predominantly enhancer regions, which it turns out are 10 times more variable than promoter regions. So there's an order of magnitude difference between the variability of enhancers and promoters, and they're also much more enriched in methylation PTLs compared to promoters that are depleted in methylation PTLs, pointing to a central importance of regulatory regions that are acting at a distance in this. You can also see that when you start correlating these enhancer methylation values with phenotypes. So if you try to classify patients according to whether they are AD or not, based on the methylation level at different regions of the genome, you see that enhancer methylation is by far the most predictive, whereas promoter methylation is actually much less predictive. And the reason, again, is that enhancers are perhaps more biologically meaningful than promoter regions. You can also see that they're much more enriched than if you use all of the covariates alone, or if you use SNPs, or if you use ApoE4 dosage. So basically, observing the epigenome is very informative in classifying AD or non-AD in these postmortem samples. But does that mean that these regions are causal? I haven't told you anything about causality yet. All I tell you is about a correlation between methylation differences and Alzheimer's case control. But you can actually bring genetics in very interesting ways. You can basically ask, do these methylation differences pick out regions that are also containing genetic variants? So if I rank all of my methylation probes according to their association with Alzheimer's, and I ask, are the genetic variants that are associated with AD enriched in these top-ranking probes? The answer is a very overwhelming yes, compared to many, many randomizations here. And that suggests that these methylation differences may not be causal, but they're at least co-occurring in regions that are causal. So that's already a very good sign. But we can go a step further and ask, is the methylation an intermediate phenotype on the way to disease, or is it simply independently affected by both genotype and the disease variant itself? And the way to do that is to correct for this genotype-methylation relationship that we've learned in our methyl PTLs and see if that correlation increases. It basically means that it was that it was not a causal phenotype, but if it decreases, it would suggest that this is a causal phenotype. And indeed, we do see a decrease, indicating that methylation is likely to be a stable intermediate in between the two. And we can do that at a much larger scale. We can basically impute methylation level across very large cohorts. So basically, we can learn this G2M relationship in a small cohort and impute methylation in a much larger cohort, and now start asking what is the relationship between imputed methylation and phenotype. Why are we excited about imputed methylation? First of all, it is the genetic component of methylation, which is very interesting. It's not, you certainly get that causality back. You also have, of course, a much larger cohort than what you would get otherwise. So that allows you to get increased power and sub-threshold signals. We can basically build these linear models, and that allows you to apply them directly to the summary statistics without access to the full genotypes, and start identifying new regions of the genome that are not associated at the genetic level, but are associated at the imputed methylation level. So how is that possible? Because imputed methylation is genetically driven. This is possible because there are multiple genetic variants that together contribute to this methylation imputation signal that would not be visible at the genome-wide level, just simply because testing all combinations of genetic marks is impossible statistically. But if you have that intermediate, you can actually start coalescing together multiple genetic signals in the same region that are independent of each other. And we can also start dissecting different phenotypes. And what we find is that from all of these methylation marks across the genome, there are different sets of marks that actually correlate with different phenotypes. If you look at neuritic plaques, neurofibrillary tangles, neuroinflammation, or cognitive decline, they're actually associated with different subsets of these methylation differences, which is kind of interesting. The same overall phenotype of Alzheimer's is now split up into multiple subphenotypes, which are in fact targeting different regions of the genome. What do these regions do? Well, first of all, they play different functional roles at the gene ontology level. So basically, different combinations of phenotypes are in fact enriched for different functional classes. And they're also enriched for different tissues in which the corresponding enhancers are active. So basically, what that suggests is that there's multiple phenotypes can be split out into subportions of the genome that they affect, and that these subportions are in fact playing different biological roles, enabling us to now take a complex trait and dissect it into its simpler components. Now, we've gone through all this trouble. We've basically, characterized the epigenomic landscape. We've identified disease-relevant tissues and regulators. We've identified target genes. We'd like to actually do something about them. And the way that I think about them is that it doesn't matter how you discover a particular mechanism. What you'd like to do now is manipulate that mechanism, regardless of whether a particular patient has the genotype that was associated or not. Because all of us have these common mechanisms, even though only some of us have perturbations in those mechanisms. So genetics gives you an entry point to figure out what to even start manipulating. So let's do that. Let's actually start manipulating these regions. So these are the 6 tenets that I mentioned earlier. We'd like to have, from a region of association, what is the tissue in which it acts, what are the target genes to which it acts, what are the causal nucleotides that are driving the association, what are the upstream regulators, and what are the cellular and the organismal consequences that result from this. So I showed you this on the first slide in the context of FTO. We're now going to dissect it in the context of FTO again. So this is joint work with Melina Klausencher, who's going to be starting out as an assistant professor at the Harvard Medical School. And this was just published last August in the New England Journal of Medicine. So obesity, I don't need to tell you how important it is. It affects 500 million people worldwide. In the US alone, it costs more than $200 billion a year. And I already introduced to you the strongest genetic association with obesity in this FTO region. So this region contains 89 different variants. It spans 50,000 nucleotides in the first intron of this FTO gene. So it was very natural to assume that FTO might actually be the target gene. And indeed, a lot of researchers focused on the FTO gene itself as a potential target. They found that FTO is expressed in many tissues, including the brain. So it made a very nice picture how changes in this regulatory region would affect expression of FTO in the brain and would somehow affect your appetite, your, whether you want to exercise, and, ultimately lead to obesity. But this story did not quite pan out. If we look at the epigenomic map that I showed you earlier, across the 127 tissues for this region, we find this massive regulatory signal for enhancer activity, not in the brain, but in the mesenchymal stem cells that give rise to 2 types of adipocytes. These are the precursor cells that can differentiate into either white adipocytes, which are the bad fat, which contains all of the lipids that make us chubby, or they can choose to differentiate into these beige adipocytes. And all of you have heard about brown fat, which is a good fat. It's rich in mitochondria. But brown fat is actually much more closely related to muscle developmentally, whereas beige fat has a lot of the thermogenesis properties of brown fat, but it is more closely related to these white fat cells. So what we found is that this precursor acts specifically in the cells that make that developmental decision. So that allowed us to now start focusing on adipocytes as a potential target tissue for the subsequent analysis. The next step was to identify what are the potential target genes. And you can see here this very beautiful insight that was first showcased by Smemo et al. In Marcelo Nombrega's lab, that basically showed this very large-scale high C region, this very strong three-dimensional block that sits together that contains the FTO locus and a bunch of other genes on one side and a bunch of other genes on the other side. So there's 8 potential genes that are co-localized with this particular genetic variant, and any of them could be the target gene. And this is actually a 2 megabase block. But simply showing co-localization is not enough. What we need to show is that the genetic variant is specifically affecting the expression of these downstream target genes. It's a great hypothesis generation, but it doesn't give us indeed a phenotype. So we went and gathered a cohort of 20 homozygous risk and 18 homozygous non-risk individuals, and we asked, is there genotype-dependent expression? And we tested all 8 genes, 2 on one side and 6 on the other side of the locus. And what we found is that there's 2 genes specifically, IRX3 and IRX5, whose expression changes by a factor of 2, specifically in these adipocyte precursor cells. These genes are sitting 500,000 nucleotides and 1.2 million nucleotides away from the variant itself. So the other thing that's quite notable is that the risk allele shows a 2-fold increased expression. So it's a gain of function. It's something that you typically don't get with protein variants. You usually get loss of function with the protein variants, but in this particular case with the regulatory variant, it just increases the expression of the genes 2-fold. So what do these genes do? Well, we'll get to that in a second. First, what are the causal SNPs? So we basically looked at regulatory motifs that are enriched across GWAS, as well as evolutionary conservation of these regions. And what we found was that there's this particular SNP that changes an AT-rich motif and disrupted by inserting a T in this AT-rich interaction domain, arid motif. And that particular SNP is both overly conserved across species, as well as occurring in the domain of multiple interacting motifs. So to make a long story short, ARID5B, among all possible candidates, was the most highly expressed in adipose tissue. So we basically followed it up, and we got lucky. And what we found is that ARID5B normally binds this AT-rich motif to repress enhancer activity and thereby repress IRX3 and RX5. But when that motif is disrupted, the repressor no longer binds, and both the enhancer and the 2 target genes are de-repressed. And we basically conducted a cis-trans conditional analysis showing that you get successful repression of IRX3 and RX5 only when you have the intact nucleotide in cis and the intact regulator in trans. But if you disrupt either by an siRNA or by the risk genotype, you basically end up with de-repression of both IRX3 and RX5, as well as activation of the enhancer, which is normally shut off in the non-risk individual. So what I've told you about is now the circuitry of that locus. I haven't yet connected it to obesity. I've told you that this particular region acts in pre-adipocytes, that the causal variant is RH1421085, this T2C disruption in an AT-rich motif, that the targets are sitting 1 million nucleotides away, IRX3 and RX5, and that the upstream regulator sits in other chromosome altogether. But altogether, we have a beautiful circuit, but we still have no connection to obesity. So how do we get that? We basically can start with the target genes. We can basically look at what are the processes that are co-expressed across individuals with changes in expression of IRX3 and RX5. When they change in expression between one person and the next person in adipose tissue, what other genes change in expression in concordance? And what we found is that these lipid metabolism genes are correlated, and these mitochondrial function genes are inversely correlated with change in expression of IRX3 and RX5. And what we find is that the risk individuals are in fact showing changes at the cellular level that mirror these expression differences. The risk individuals show higher adipocyte size and actually significantly higher adipocyte size and significantly lower mitochondrial activity, suggesting that there's a shift from energy dissipation in the mitochondria to energy storage in the risk individual. So That gives us a mechanism that basically says that perhaps what's happening is that there's a developmental shift between a white adipocyte program and a beige adipocyte program that happens because of that one SNP in the FTO locus that causes IRX3 and IRX5 to be de-repressed, which then turn on thermogenesis programs and turn off thermogenesis programs and then turn on white adipocytes. So how do we now utilize that circuitry to actually manipulate the disease phenotype? So we can now go in. We're open for business. We have the target genes, we have the upstream regulator, and we have the causal variant. And we can intervene at any one of those knobs. We can overexpress or downregulate IRX3 and IRX5, and we predict anti-obesity phenotypes with knockdown and pro-obesity phenotypes with overexpression based on this regulatory circuitry. Similarly, for knockdown and overexpression of the IRX5B regulator, we predict pro-obesity or anti-obesity phenotypes, and with the specific nucleotide alterations, which we can now carry out using CRISPR-Cas9 editing, we can actually shift, in the context of the haplotype, a single nucleotide between C and T, and basically see how it changes in the risk and the non-risk individuals. And the answers were remarkable. It basically functions like a switch. If you change a single nucleotide here, two genes, 500 kb and 1.2 million nucleotides away, change in expression by a factor of 2. And if you switch that nucleotide back from T to C back to T, you basically see this re-repression of both IRX3 and IRX5. I mean, that's remarkable. It basically suggests that long-distance regulation is extremely precise. But even more remarkable is the fact that if you look at the thermogenesis phenotype, which is basically the relative oxygen consumption rate in presence of hyperterenol, which is a brain agonist for thermogenesis, you basically see that you can take a risk individual who's unable to thermogenize and take the fat cells that are simply, storing up all this energy because that pathway is broken, and change a single nucleotide out of 3.2 nucleotides in the human genome, 3.2 billion nucleotides. I mean, I don't know how much more precise it gets for precision medicine, but you basically change a single nucleotide, and what you see is restoration of the thermogenesis pathway. You see the 7-fold increase in thermogenesis, which is extremely highly significant, by altering a single nucleotide. So that's at the cellular level. What about at the, and that's for the nucleotide itself. We can also show that these changes are in fact mediated specifically by the two target genes, IRX3 and IRX5. So overexpression or knockdown of IRX3 and IRX5 are shifting thermogenesis in both risk and non-risk individuals exactly as you would predict, and they have no effect on the opposite phenotype, on the opposite genotype. And lastly, that's at the cellular level. At the organismal level, we can go into mice and create a dominant negative form of IRX3, which is expressed in adipose tissue. And what we find is a 50% loss in fat mass ratio. You find that these mice are simply unable to gain weight in a high-fat diet. So you introduce a high-fat diet. These mice are smaller to start with, but they show no increase in weight when the normal mice are, in fact, simply gaining weight on a high-fat diet. So these mice are losing 50% of their body fat and unable to gain weight when you put them on a high-fat diet. And they don't change their appetite. They don't change their exercise. And this is also happening in thermoneutral conditions. But what's remarkable is that they're simply dissipating more heat. These mice are just simply burning energy, both when they're active and when they're sleeping. So it basically suggests that by figuring out the circuitry and by manipulating the circuitry, we can actually go and change the metabolism of these fat cells, and that has an organismal effect in the case of mice. You can basically shift between white and beige function using it. And what I want to really point out is that we're at a very early stage here. So there are thousands of these non-coding variants that are disease-associated. This is number 14 in the p-value list for the non-coding variants out of several hundred. It's more than 1,000 right now. And we have the potential to actually gain tremendous insights by doing this systematically, disorder after disorder. But we need to do this in a much higher throughput. So in the last part of the talk, I'm going to talk just about a couple of technologies that we're using to scale this up by several orders of magnitude. The first technology is actually utilizing high throughput enhancer activity assays where we can test the effect of individual variants. And we're doing this in collaboration with Dr. Nicholson and Vijay Sankaran over at the Broad Institute, and Jason Ernst, who is now a professor at UCLA and used to be a postdoc in my lab. So what we can do is actually start manipulating these in specific cell types and watching their effects. We can do this at much higher throughput. We can actually predict individual nucleotides that are relevant in any one of these disease regions. If you look at across hundreds of these regions, you can basically see that there's very strong agreement between the specific driver regulatory motifs that we predict and where these occur. We can get a bunch of insights as to the regulatory circuitry, which I'm not going to have time to cover. And we can start predicting computationally the effect of both common and rare and somatic mutations, because certainly for common variants, we can test them systematically once and for all. But for rare variants and somatic variants, we don't have that luxury. So we have improved a lot on the computational models that allow us to now exploit genome-wide experiments to predict the effect of individual mutations. And the last thing that we're doing is actually multiplexing the validation experiment by basically taking a large number of embryonic stem cells that are fully haplotype resolved, differentiating them into different lineages, and then testing specifically for allelic activity in the heterozygous loci for each of those individuals to basically see where do the genetic variants manifest themselves at the cis level. So we just got funded as of yesterday for carrying out this systematically in the context of schizophrenia in collaboration with Jana Mitchell and Kevin Egan at the Broad Institute. To basically take stem cells, differentiating into several different types of lineages, and then observe where do these genetic variants manifest themselves, and then carry out in 384-well format systematic reporter assays to basically look at the cellular changes that are happening in the context of these genetic variants that are predicted by our circuitry. So that's what I want to leave you with, that we can systematically characterize the epigenomic landscape, utilize that to identify disease-relevant tissues and regulators, and that can lead to some very strong surprises in the case of Alzheimer's disease, for example, pointing to a central role of immune and inflammatory drivers. We can use that to identify new genes below genome-wide significance thresholds, using epigenomic signatures that we train at the genome-wide significance level, using convergence of these scattered rare and somatic mutations into a common set of target genes, and using epigenetic variation across individuals to identify new regions and subdivide the different phenotypes of AD. We can now go into these regions and utilize our predictions about the circuitry to identify the specific circuits that one should manipulate and manipulate these circuits in human cells as well as in mice. And we can scale this up, basically use massively parallel reporter assays. We can basically use computational models to predict these individual driver SNPs. And we can use iPSC and ES differentiation and 384-well format to basically speed up the process of validation to go to a small set of really high accuracy and highly precise hypotheses for the disease variant. None of this work would be possible without an incredible set of collaborators. So for the Roadmap Epigenomics project, Anshul Kundaji was one of the leads. He's now a professor at Stanford. Varo Meliman just took a new position over at UW. Jason Ernst is now a professor at UCLA. Xinchen Wang is still stuck with us, but he's graduating soon. He did all the cardiac threshold work. Matt Eaton is now a key player in biotech, a startup in Boston. Andreas Penning just started his position at CMU as an assistant professor. This was joint work with Lisbeth Haneska in Ligwei Tsai's lab. And then for the FTO story, Melina Klausner starting her position at Harvard. Richard Salari just joined a new cancer startup. And then we have this amazing collaboration for high throughput validation. So I'll stop there and take any questions. And I don't know if I made this clear, but I'm looking for postdocs. So if any of you guys are finishing up, come over. Please.
That was great. I mean, Alzheimer's, diabetes, all the other things that you kind of are underpinning on you thinking is fantastic. It's sort of force thanks. I'm thinking about allele-specific expression.
Sure.
I mean, there are two chromosomes here, and allele-specific methylation. And there must be, I mean, we've used the beer classifiers. I mean, there must be some way that you can tease out that as part of the scheme.
Absolutely. So that's exactly what we're using. So we have a paper that's actually being written right now for specifically Alzheimer's disease, looking at allelic differences at the gene level and allelic differences at the enhancer level. It gets quite complex because if you look at within the particular haplotype, so why are we using this? We're using this to basically look specifically at cis changes where the enhancer is in fact driving changes in expression of the gene at the cis level, controlling for everything else in trans because we're looking at allelic activity. If you're within the same LD block, you're kind of stuck because everything will be correlated. Any change, you know, in an enhancer will also be a change in a, you know, transcribed element. So you have to look outside the enhancer block. When you look outside the enhancer block, you're still stuck because you don't know the phasing between that allele and the other allele. So what we're doing in the context of this particular grant that just was funded is that we're looking for a set of haplotype resolved individuals, so fully phased individuals. So for every one of these individuals, we actually have the linking between the enhancers and the distal promoters across LD block boundaries, because that's the only place where we should be looking for signals. So we're starting out this experiment right now. I'll report back. What you can do when you don't have phasing is look for increased heterozygosity associated with allelic differences. You don't know the direction of effect, but you can look for over-dispersion at the enhancer SNP level, and that indeed will be fine. So basically, in the Alzheimer's paper that we're wrapping up right now, we're finding this signal of, you know, increased heterozygosity outside the haplotype block in regulatory elements that are associated with a particular allelic imbalance at the gene level. Great question.
I think I have one more, and that is that I'm thinking about this trans interaction, and one thing I loved about it is laying out the idea of the regulatory motifs that way. You know, they're probably co-evolved with the. I would think that there was co-evolution going on there. I mean, could you comment on that?
So we had an awesome chat earlier today about multiple drivers in the same haplotype, and how is that even possible? And there's a bunch of ideas that we have surrounding this. I'm happy to talk with you offline. But a very simple way to think about it is that you basically have accumulation of mutations in absence of selection. You cannot possibly select in the human population for multiple consecutive mutations driving towards the same phenotypic difference in a particular locus. This is, you know, human population is very small. Number two, driving towards a disease locus suggests a strong environmental change, and that kind of stuff is possible. For example, in BMI, where you can actually sort of have this, you know, thrifty gene of basically looking for increased energy consumption from a scarce set of resources, which only changed in the last few decades, leading to the huge epidemic of obesity. But you know, the cases of strong selective pressure are, you know, I can count them into my two hands, basically. There's very few of those. And it's unclear that they're driven by multiple consecutive mutations rather than a single haplotype that was selected. How do you get multiple drivers in the same haplotype? Well, one possibility is that you basically have multiple drivers that are independently changing things for or against the disease. And GWAS is basically selecting, setting a threshold for multiple of those drivers in the same direction. So they're occurring by chance. Some are activating, some are repressive, but we're looking specifically at the disease loci, and these are more likely to have multiple variants driving in the same direction. Does that make sense?
Thank you.
Thank you.
Other question?
There's one, too.
Might be pessimistic, but one of the things I've noticed about the epigenome in particular, this small space that we tend to live in, of the interaction between the epigenome and the three-dimensional, four-dimensional structure of the nucleus, is that the people who live inside this community are very enthused about it and have been doing proof of concept studies on a lot of different potential applications that have a lot of translational potential to a lot of different areas. But there's a tendency for people who live outside of this area to be skeptical about the whole concept and to think sometimes that we're living in the clouds. And the question is, how do we convert and translate?
So thank you so much for bringing this up. So there's a lot of misconceptions surrounding the epigenome. So I call it epigenomics, not epigenetics. And the concept of epigenetics is very charged. It basically says that it's about inheritance, that you can have an environmental change which causes a epigenetic difference, which then gets passed on without any genetic mutation from generation to the next. And that's very controversial. But everything I showed today, I did not use a single time the word epigenetic, because I do not make any implications about inheritance, and I do not make even any implications about causality until our causal inference models are showing this. I showed epigenomic alteration. And what does that mean? That basically means that I'm looking for regulatory annotation. So you don't have to even use the epig word at all. You can simply say regulatory annotation, regulatory alterations, regulatory changes, whether or not they're causal or simply indicative of an underlying difference in activity. I'm agnostic. All I care about is that they're a way to measure activity outside genes. And that's what we're missing. What we're missing is the ability to characterize epigenomic dynamics. And in one of the very first slides that I showed you, and I'm going to try to find the home button here. I basically showed you these, epigenomic maps across a very large number of cell types. With only gene-centric views, we would have, this tiny little exon here as the only thing that we'd be able to measure. Epigenomic profiling lights up the genome. It basically allows you to now paint activity differences in specific regions. And this is extremely important because the non-coding variants are acting predominantly by a 93% to 7% difference in non-coding regions. So common variants are predominantly acting in non-coding regions. So it's not like some fringe element of our genetic community that we're facing here. This is like the 93% of GWAS. The genetic community cares about signals, and that's extremely important. But people like Liz know that the signals is the beginning, not the end. And from there to mechanism is a huge path. And that's where we are. And in that huge path, the message is out that non-coding is the key to understanding GWAS.
Last question?
Manola, she gave me special permission for this one because it's.
I would have given you two.
But it really is a general question. I was fascinated by the idea that you gave this whole talk without mentioning two key ideas.
Oh,
One is the loops, the enhancement promoter loops.
Sure.
Laid out
Yeah.
That are part of the mechanistic story.
Yeah.
And then the others are the topologically associated domain.
Yeah.
And how they, in the sense of either within the chromosome or allele or across.
Yeah. So there were two small mentions, but you're right. We're still at the very early stages of the 4D nucleome, basically. And there's like massive efforts to complement that. But basically, these data sets are very scarce. In this particular case, we were very lucky because we have this high C evidence here that allowed us to find this large domain. But that was not the end of the story. That was the beginning of the story, that any of these could be candidates. And then the other place where I used it was in the context of these regulatory plexus of the gene, where we're basically using these high C interactions to basically link together. Yeah, and I think you're absolutely right. We have to think much more about this long-range connectivity. And we're using 3 lines of evidence. High C is 1 line of evidence. That's physical interaction. Correlations is the 2nd line of evidence. That's basically this enhancer lights up in the same tissues that this gene lights up, so we can connect them based on activity correlations. And the 3rd is, of course, EQTL. So I think we should think of high C very, very strongly, but in the context of these other 2 lines of evidence, because they're complementary. They don't always agree, and the reason they don't always agree is because they measure different things: genetic links, physical links, activity links. I agree with you completely.
Thank you.
All right, thank you.
Great.
Thank you very much. Thank you very much.
Amazing. That last bit. I loved it.
Oh yeah, that's good.
Yeah.
Hey, how's it going? Thanks so much for your last minute. You saved the day, man. And I That is awesome.